Hi everybody, DrBiology here, and this is part 7 of my biological molecules unit on DNA replication. So DNA replication is due to something called semi-conservative replication. So before a cell divides by mitosis or meiosis, the DNA in the nucleus is copied or replicated. So it was Watson and Crick who worked together to confirm the double helix structure of DNA in 1953, for which they received the Nobel Prize. And they came up with a model which DNA might be replicated, called a semi-conservative DNA replication model. I'm not going to go further until I do mention Rosalind Franklin. She had a really important part in actually helping Watson and Crick with the double helix structure, doing work on X-ray crystallography. So if you want to learn more about that, please do check out my video, as you can see in the bookmark right now. So let's talk about DNA replication. So first of all, a portion of DNA representing base pairs is held together by hydrogen bonds. And as you can see, it's complementary base pairs, G to C, A to T. Then the enzyme DNA helicase breaks the hydrogen bonds between the bases on the two polynucleotide DNA strands, and this makes the helix unwind to form two angle strands. And then free nucleotides from the nucleoplasm are attracted to their complementary bases on each separated strand of DNA. And the enzyme DNA polymerase joins those nucleotides together. And as you can see, each new DNA molecule consists of one strand, so one original template strand from the original DNA double helix and one newly synthesized strand, and it's shown here in a different color. So you can see the red strand is the template, and then the pink strand is the newly synthesized strand. And therefore, two identical DNA molecules are formed. So key points on this are: 1, DNA helicase unwinds the DNA and it breaks the hydrogen bonds. Number 2, both strands act as templates. Number 3, free nucleotides line up in complementary pairs, so A to T and G to C. Then 4, DNA polymerase joins the nucleotides of the new strand. 5, forming phosphodiester bonds. And then each new DNA molecule consists of one original template strand and one new strand. So who came up with the idea? So who actually did the experiment to look at and prove that it was semi-conservative replication? So it was these guys here. So it's Matthew Meselsohn and Franklin Stahl. So in 1958, they came together and they tested different models at that point for DNA replication. They were at Caltech in California, a very famous university related to genetics research. And they definitively settled the question, and in the process revolutionized the fields of biology and genetics. This is what one scientist said, Judith Campbell from Caltech. The Meselsohn-Stahl experiment has been called the most beautiful experiment in biology for the elegant logic of its deceptively simple design. So what was the evidence? Well, there were 3 hypotheses before had been proposed for DNA replication. So the first one was the conservative model. It was an entirely new molecule is synthesized from a DNA template, which remains unaltered, as you can see in the diagram. So you're getting different, entirely new strands, basically double strands. The semi-conservative model, where each new molecule consists of one newly synthesized strand and one template strand. And then finally, the dispersive model. So new molecules are made of segments of new and old DNA. So that's suggesting that somehow the DNA is cut up and mixed together to produce 2 strands made of segments of new and old DNA. So what did Meselsohn and Stahl do? Well, what they did was this. They designed an experiment to determine the mechanism of DNA replication. So they grew E. coli, which is a form of bacteria, on agar plates in Petri dishes, and they contain the heavy isotope of nitrogen 15N for many generations. So all the DNA was labeled with heavy nitrogen. And then the bacteria were transferred to a medium containing the lighter isotope of nitrogen 14N for 1 or 2 or 3 generations. What they then used was an ultracentrifuge, and basically they killed the bacteria and they extracted the DNA and subjected it to density gradient centrifugation. So basically sticking it in a centrifuge and then looking at the bands of DNA. And as you can see, there's light DNA is less dense, so it's much higher up in the tube, and heavy DNA is low because it's more dense. So there we go. So DNA samples sink to a point in the centrifuge tube equal to their own density. They then made the samples visible under UV light. So this was part of their experiment. So you can see the controls. So you've got the 15N and the 14N, and then it shows the incubation of heavy cells in 14N. And you can see the first generation. So that's suggesting that one strand is 15N and another strand is 14N. And then you can see the second generation where it splits again, and you've got 2 strands of 14N and 1 strand of 14N and 15N. So these were the actual observations here. So you can see they looked at all aspects of the predictions, and as you can see, it fits with the semi-conservative model. So the results after 2 divisions supported the semi-conservative model of DNA replication. After one division, DNA molecules were found to contain a mix of 15N and 14N, disproving the conservative model. And after two divisions, some molecules of DNA were found to consist solely of 14N, therefore the second generation disproving the dispersive model. Okay, I hope you found that useful. I'm going to go through some exam questions for this section of work because Meselsohn and Stahl does come up quite a lot. I hope you found that useful. My next video, I'm going to be looking at the structure and roles of ATP. I'll see you soon.